For this installment of OMG OMx, you're going to hear from one of my favorite people because he does across the omics space. Stay tuned to learn about the impacts of what he's been working on. Hi there, Blaine. Welcome to OMG OMx. I was hoping that to start off today, you could tell us about how you see the natural world and how you had influences on this growing up.
Yeah, well, Kate, thank you for having me. Appreciate you, inviting me into the OMG OMx. I've been watching it for many of the podcasts for the last several months. So my background really is where I got interested in the natural world, and that was growing up on a farm. So I grew up on a medium to small family farm in Michigan, where we raised hogs or pigs, as well as horses and so forth. And so just growing up in that environment was really unique, I think, because one, you get biology in the husbandry of taking care of animals as well as in growing crops. And then at the same time, you get a lot of technology, right, in dealing with the technology that's used to harvest and to harvest crops and to just run a farm in general. And a lot of that's mechanical, but that's really set me up to where I'm at now, where we do a lot of biology, but we use advanced technology to answer those biological questions. So it really did start from birth for me, because that's where it started off.
So how did this lead into your undergraduate and graduate research days? How have your research interests been impacted from your time in your schooling?
Yeah. So the other thing is, my dad was a biology teacher as well. So he could always answer or at least come up with a good explanation or answer for the questions I might have as I was growing up and, around biology and so forth. But then I went on, so that influenced me greatly, that I was very interested in science and I always did well at that subject as well. And then went on to undergraduate education at Montana State University in Bozeman. And there I was majoring in chemistry with a biochemistry option because I liked chemistry, but maybe not as much as I liked biochemistry, right? I liked the squishy stuff, not just beakers and flasks. I didn't mind, again, kind of going back to my roots, being able to see biology and biological, how the biological processes are occurring, right? Which was really interesting to me. There I worked with my undergrad research advisor, Ed Dratz. And it was quite interesting because we started working on dissecting the retina out of cow eyeballs so that we could isolate the rod cells, which are the cells in your eye responsible for your night vision. And, we would take that to isolate the G protein called rhodopsin and do biochemical studies on that. And actually was where I first got exposed to mass spectrometry. They had a MALDI system where we would use that to do peptide mapping of that rhodopsin protein. And so really it was full circle, right? Going from the farm where we had pigs, we had a few cows as well, and then going to dissecting these cow eyes to isolate the protein and then start doing these biochemical investigations of how these proteins are regulated and function in our vision. From there, I went to do my PhD at Oregon State University with Professor Joe Beckman. And that's really where I got interested or introduced to neurodegeneration. And so my thesis was around ALS or motor neuron disease and trying to understand the role of a protein called SOD1 that's involved in antioxidant response, but also mutations of that are involved in development of familial form of ALS. And again, mass spectrometry played a prominent role there. They had a really great mass spectrometry center and facility, both for environmental science as well as the biological sciences, which is what obviously we were working on. And from that, I've continued to collaborate with both my undergrad research advisor and my PhD research advisor to this day. We just published a paper last year together on some of the research that we've been doing since. And my thesis advisor even helped develop new mass spectrometry technology that's made it into the current instruments, which was kind of fun to be part of, or to be around when they were doing those developments as a graduate student. From there, I went to the deep south, all the way to Australia, to the University of Melbourne. Really, it was at the Mental Health Research Institute, which later became the Florey Institute of Neuroscience, which is on the University of Melbourne campus. And there I was working for my postdoc advisor, who is Professor Colin Masters. And that's where I really got into Alzheimer's disease research and a bit of Parkinson's as well. Colin is known for his research on amyloid beta, the peptide that causes Alzheimer's disease. And there I was working and doing a lot of protein biochemistry and protein purification, kind of similar to where I started as an undergrad, purifying proteins from natural sources. In that case, it was cow eyes. This time we were moving up the food chain, so to speak, working with human tissue, trying to purify different variants of the amyloid beta peptide and other proteins as well.
So recently I had the privilege of sitting in on one of your group meetings. So maybe we can pull this story up to now and tell me a little bit about your motivation in guiding your students through the bigger picture and how you make it relevant to them and not just their individual pieces.
Yeah, so that was great having you here, and you get kind of experience the lab and meet the people here doing the work. And I think my motivation and my philosophy, I suppose, with working with undergraduates or even high school students, all the way up to PhD students and postdocs and so on, really is about understanding the foundation and the fundamentals, and also getting the big picture, right? So often on a project, we're working on one small piece, right? Maybe it's, as an analogy, right, building a rivet or something like that, which isn't very exciting maybe, and you don't know how it fits into the bigger picture. But when you're building a rivet and that's going to go together to make the Eiffel Tower or some other much larger project, it gives you a lot of motivation and understanding of why you're doing it, right? Versus it just being mundane. And so we have two philosophies. One is knowing the bigger picture. What were we trying to do? We're trying to cure Alzheimer's disease. We're trying to cure Parkinson's disease, cure ALS. We have a big focus on trying to develop new diagnostic tools because they're difficult diseases to detect and diagnose. And knowing that whatever it is you're working on might not be the assay at the clinical bench, but it's part of that process and part of building that tool, or in this case, that tool or that cure or that understanding that's needed to reach that.
That's fantastic. And I think a good thing that you've done that's really empowering is your lab works in multiple omics spaces for your research. Tell me a little bit about how the community sees this and the breadth of your investigations. And do you think more researchers should stray out of their omics lane?
I think so. In general, I would say the omics community are very open to it, right? And I think in general, we all want to do it, and sometimes it's about bandwidth and or opportunity to do it. So we're really fortunate here at Emory to have the bandwidth and the opportunity to do multiple omics. And I think everybody, including myself, right, we all realize it's really important to have the full picture, right? If we're studying proteomics, which, my background in protein chemistry and protein biochemistry, that's my bias, right? But of course, proteins don't live in isolation, right? There's all these wonderful things like lipids that make up the membranes that do cell signaling. Then there's, the metabolites, the RNA, DNA, all of that comes together to really paint the full picture of the pathological process that are occurring in neurodegeneration. And I think, focusing on one is great, but to build a more comprehensive picture and unbiased picture, you need that multi-omic space. You need to know what the lipids are doing. And like our project that we're working with the Michael J. Fox Foundation with at the moment, looking at biomarkers, we're bringing all of that together. So we're looking at metals. What's the role of iron and other elements in the disease? The lipids, as well as the metabolites, as well as the proteins. And what's been fantastic in that is, as we've collected that data and done our analyses, is that we're starting to see common themes, right? So often we might do just proteomics, and we do our network analysis and try to interpret what that data says. It says, oh, it's pointing to biological process X, whatever it might be, apoptosis, let's say. But when now we're able to do the lipidomics, it's also pointing to that same biological process. And so it starts to really build a more complete and more comprehensive picture of the biological processes, and gives you more confidence that, oh, the changes we think we see that we see in the proteins are also reflected in the lipids, and also reflected in the metabolites, also reflected in the metals, for example. Really gives us greater confidence in it, and just a more complete view of the tapestry, right, that's occurring, that's describing this disease.
So one of the key ideas in biology and some of the biochemistry that you've studied is around structure-function relationships. Tell me how this is critical to your omics-based research.
Right. Well, I think it kind of goes back for me, and probably for everybody, the whole reason why we even have proteomics really is born from the idea of trying to understand the role of proteins, because we know that proteins are the molecules that are doing most of the work, right? They're the molecular machines, right, for the most part. And in general, we're trying to understand protein structure, protein function, right? And that's for drug design, also understanding kinetics, but then also localization is important, right? Is it in the nucleus? What cell type is it in? So in general, proteomics, we're trying to understand as much as we can about the protein, not just all of the proteins, but we want that complete picture of how all the proteins are working together. And at the moment, proteomics is an excellent tool at telling us how much of a protein is there, right? And it's very highly plexed, right? We can measure nearly all the proteins that are expressed in a cell at any given time nowadays. But that regulating protein concentration, or the abundance of a protein, is only one way the cell can regulate its response to its environment. And we know from post-translational modifications and other cell signalings that's too slow, right? Changing the protein concentration takes a long time. And we know that the protein function, so that trying to get at that structure and the enzymatic activity, its protein-protein interactions, those are the regulations that are kind of the next frontier, right? Trying to understand that and see how a cell responds in real time, where it isn't just a change in protein level, I think is where we're trying to go. Yeah, I think in general that's what we're after, is trying to understand how proteins work to give us cellular function.
Neurodegenerative diseases have been part of your research focus. How has being in the clinical research space affected your view and your motivation?
Yeah, that's an excellent question. I think, well, in general, definitely been a positive effect. And, I think all researchers we're doing work. Sometimes it's blue sky, just to try to understand how something works, right? That's true. But often there's a why. Well, why are we trying to understand that? Well, because we want to help a disease or help improve the quality of lives of somebody, extend lifespan, whatever it might be, right? But there's an actual real human impact. And for us, working at that edge between the clinic and the basic research is interesting, because it brings unique challenges as well, right? So we have, I guess, terms we use in my lab, which are biological biomarkers. So those are the things that change, that are really interesting, but you couldn't use them at the clinic. You couldn't use them as a diagnostic biomarker or another type of clinical marker. And so that's a distinction, I think, for me anyways, was interesting to experience, right? Is seeing what a diagnostic marker needs to look like is very, very different from the biological biomarker, which tells us about the disease or tells us about the process that's occurring. And the other part that's been really beneficial and advantageous is being able to work with clinicians who are seeing the patients and having a much better understanding of maybe the problems that they're facing, right? They're kind of like the end user, right? You have the patient and the clinician who are the end users. So how would they use it? What are things that they see as a challenge, right? From my side of the academic research, I'm thinking, oh, that diagnosing X or understanding Y is the really important thing. And then you go and talk to a clinician, they're like, we can do that, or that's not really that important. Would be really impactful for a patient is being able to understand this, right? Knowing when to give, when do I need to take my medication and how often, what dose do I need to be at? So which, for us, we could potentially develop markers that could monitor that, right? So that's been really beneficial. And of course, being able to get access to clinical samples is also a huge plus or benefit, right? We can't always work with model systems.
So maybe let me throw some numbers at you, and you can share if you feel any personal impacts or even just emotions around the work that you do. So more than 10 million people worldwide live with Parkinson's disease, with nearly 90,000 people in the United States diagnosed each year. In addition to that, over 40% of the global population suffers from some sort of neurological condition.
Yeah, I mean, those are huge numbers. And if you've ever known anyone to have Parkinson's disease or Alzheimer's disease or any of these neurodegenerative diseases, right? We know they're an awful condition, and you feel empathy for them. You feel fear. As I'm getting older, I hope I don't. I hope I don't develop that, or my loved ones don't develop that disease. And so that's definitely a driving force. Knowing that the research we're doing could have an impact on tens of thousands of people, or potentially even millions of people worldwide, is helps keep you get out of bed in the morning, right? Keeps you going. And when you feel bad, you're like, my day is never as bad as somebody who's having to live or experience those diseases, right? And it's wonderful when you meet patients how upbeat many of them are, right? They're positive. They want to help. Um, and I think in general, it helps you just appreciate what you have.
With your work with the Michael J. Fox Foundation, has that put you into those spaces more than maybe the average clinical researcher would have the opportunity to engage with real patients, real people living with this?
So my work with the MJ Fox hasn't done that necessarily, as far as that there isn't a program, or maybe there is, but I'm not part of a program where we meet regularly with patients. Usually that comes from our local contacts and local groups, which MJ Fox help us network with often. And then also just from experience, right? We have had people who are family members or are friends of family members who've developed these diseases over time. And so that's been our main contact with patient populations in general, or people with the disease, right? And it was actually, I still clearly remember the first meeting I ever went to, and this was as a graduate student with Joe Beckman, and he took us all, the whole lab, to an ALS support group meeting, right, where he gave a talk on what he thinks is going on. And I think that was really impactful, right? For me, it was by a long shot, because that abstract thing, right? We always start off our introduction about describing what the disease is, right? It's like, ALS is, this many people per 100,000, or PD or whatever, second most common neurological disease, and it's described by tremor and so forth, right? Whatever these things are. But it's very different to meet people and see what that effect actually is, and see what, I guess, the impact on a person's life, right? That's a very different introduction when it's, visceral right there in front of you. And so that was really powerful to go, hey, what I'm doing is quite small and may not translate to that, but it can be part of that bigger project, right? That eventually leads to a treatment or something that can have a positive impact on people who are suffering from these diseases.
I'm glad you brought up ALS. Maybe you can tell me a little bit more about how Alzheimer's disease, Parkinson's disease, and ALS are similar to each other, and maybe even some of the differences in how you have to approach research in a orthogonal way.
Yeah, so those are kind of the big three, I think, that are probably some of the most researched neurodegenerative diseases, and then also they're some of the most common ones as well. Obviously, Alzheimer's being the most common, and PD being or Parkinson's being second most common. But they all affect very different areas of our nervous system, and have very different clinical presentations or very different symptoms from one another and different rates. So ALS is a disease that's the most rapid of all of them, right? The average time from diagnosis for the disease to death is, in that range of 3 years to even 5 years, right? So it's quite a rapidly progressing disease. Whereas Alzheimer's disease or Parkinson's disease, we know from pathological studies and imaging studies that is a disease that has taken 30 years to develop. Now, that could be the case in ALS. We just don't know that at the moment. But it takes 30 years to develop, and then after somebody is diagnosed, individuals can go on to live with those diseases for 5, 10, for decades, up to decades on that. So that's a very big difference, I suppose, in both the pathological processes maybe that you're looking for, and then on top of that, they affect completely different populations of neurons. So ALS is hitting your motor neurons, the neurons that are used to move your muscles, whereas Parkinson's are hitting neurons that are in your substantia nigra, and then Alzheimer's disease is hitting neurons basically throughout your entire cortex. And so obviously, when we're doing our investigations, we're looking at different areas of the brain. So you can't look at just one and try to understand all three. However, there are common themes, right? There probably are common ways that neurons die. And we think in particular with PD and in ALS, we've at least shown this in animal models, that some of the therapies that we've been working with that work in the ALS models also seem to work in the Parkinson's models. And so there may be some common themes on how those two diseases occur. And that's an area of, keen interest and research for us at the moment, and others, right? That we aren't the only ones to think that or to notice the similarities between Parkinson's and ALS, at least in how the neurons respond and how the neurons die.
So in recent years, it was discovered that alpha-synuclein seed amplification assays can be used as disease biomarkers from cerebrospinal fluid. What are the impacts of the biological biomarker discovery that you're doing, and how long would it take to actually translate into a disease biomarker?
Yeah, so the recent reports on the seed amplification assay, basically picking up these pathological forms of alpha-synuclein from the spinal fluid as diagnostic markers, has been a very an exciting development, and I think potentially a game changer in the disease. So Parkinson's disease is behind something like Alzheimer's. Alzheimer's disease, now we've gone through this process of having no biomarkers to having only biomarkers that are for imaging, to having only biomarkers that are in spinal fluid, to now having multiple blood biomarkers that we can use to diagnose Alzheimer's. Parkinson's disease is behind that. We don't have biomarkers until this one, right? This is showing up as a biomarker. It's in this, at the moment, it only works in spinal fluid, which is an invasive, potentially an invasive way to get, your biological sample to run the test. But of course, it's worth doing, and it's working, and it's going to help us design better therapies or therapeutic trials. And that, because still one of the biggest challenges, or a major challenge in Parkinson's disease, is misdiagnosis, right? And we still know that 10% to even up to 20% of the people diagnosed with Parkinson's disease at autopsy don't actually have Parkinson's. They have a different type of disease. So having a biomarker that could detect that is really powerful for testing new therapies.
So before I get to the last question, let me just say thank you. And I've learned so much in every interaction that I have with you. And I'm just so motivated myself to learn more about this space and understand the research that's going on. So my last question for you is, you have a strong family connection in your lab. So tell me a little bit more about that and what it's like working with your wife, Anne.
Yeah. Well, and thanks for that comment too, Kate. I really appreciate it, and I really enjoy working with you as well over the years. Um, and yeah, working with Anne, who is my wife as well, right, has been a lot of fun. And I think in science in particular, um, it's as much like maybe being an artist, right, where um, you really want to understand how something works and how the world works. And so you're thinking about it all the time. It's not on and off. And having a partner that is similar minded and we're both working on that same problem, right, or similar problems has been wonderful. And the other part is that where I might bring some, what would it say, maybe like some mess or clutter to a situation or to an actual physical space, and is there to help streamline it and so to improve it and also keep things within the rules, right, and really pull out what is required to make a process work. And so yes, sometimes we struggle talking about work all the time, right? Like talking about it at home, and our kids roll their eyes and they're like, okay, enough about alpha-synuclein today. Right? We have high schoolers who've heard of alpha-synuclein, and but in general, it's wonderful, and it's really part about having a team, right? Even though I have a singular interest or singular interests, um, and I'm working on that one thing, it's really you need a team to be able to tackle these larger problems, and that's what's about having a bigger lab, about having students and postdocs and so forth that are in there. And Andrew just really helps get everybody trained on the same page, taking care of the fundamentals and those little things so that we can do these big experiments.
All right. Well, thanks again, Blaine. And I look forward to the next installment of all the things that you've accomplished.
Wonderful. Thank you. Have a wonderful day.
I hope that today's installment has helped you to see the goal of OMG OMx in bringing you a closer personal connection to the research that many people are doing.